So we learned how the action potential starts and how it moves down the axon, and this slide right here shows you how it ends, if you will. So what we're looking at right now is a synapse. Okay, you see this axon terminal called the pre-synaptic terminal, pre-synaptic, and this is the post-synaptic terminal, or a dendritic bulb from a dendrite of another cell. So here's the end of one axon, and here's the beginning of a dendrite. So we have this gap called the synaptic gap, but please note pre and post because it's kind of important. So the presynaptic terminal is going to release its neurotransmitter, and it's going to bind these chemical gated ion channels of the postsynaptic cell. So here's the action potential coming down. Okay, so sodium in and potassium out. It's jumping. Here to see the myelination right here. So it's going to jump in. The very last thing it does is the action potential stimulates voltage-gated calcium channels. Calcium is another ion I told you we'd talk about. And there's that calcium channel, that voltage-gated channel. And when it changes the membrane voltage, the channel opens due to the voltage gate, and calcium rushes in. We're just going to assume there's calcium rushing in. Turns out there's calcium inside and outside of the cell, but it does have a driving force to move into the cell. It moves into the cell, and in this axon terminal are these vesicles, and the vesicles hold the neurotransmitters. They're made in different parts of the cell. The proteins are made in the nucleus, of course, and then they're shuttled down. Here's some cytoskeletal elements we talked about. They're shuttled down and packaged constantly. So they're packaged, and once they're packaged, they're hanging here until calcium comes in and untethers them, cuts them loose so they can merge with the membrane. And when they merge with the membrane, since they're made out of phospholipids and the membrane is, they just blend right in, and they dump their contents into the cleft. They dump their neurotransmitters, whether it's acetylcholine like we talked about, or norepinephrine. It could be serotonin or dopamine, or who knows. Comes across the cleft, and then it binds to the appropriate receptor and has the influence on this cell and starts this potential action potential cascade all over again in another cell. If this is an excitatory neurotransmitter that's being released, it's going to stimulate, it's going to cause a depolarization here. It's going to allow sodium to move in, and it's going to start moving that threshold phase, the first phase of the action potential. So the charges that occur on the postsynaptic cell are called synaptic potentials, changes in voltage along this membrane. And we're going to talk about the difference between an excitatory postsynaptic potential and an inhibitory postsynaptic potential. This one's excitatory, brought on by excitatory neurotransmitters, depolarizes the cell and moves the cell closer to firing an action potential. And this is inhibitory, brought on by inhibitory neurotransmitters. It inhibits, hyperpolarizes this region of the cell, and moves it further away from firing an action potential.